Let us map the relevant neurobiology more explicitly. A useful sequence is this: stimulus, dopaminergic activation, action, outcome evaluation, and learning. Once a stimulus is encountered, the brain assesses its salience. In simple terms, it asks whether this stimulus is important. Structures such as the amygdala are involved in emotional salience. The prefrontal cortex is involved in evaluation, judgment, planning, and control. If the stimulus is appraised as meaningful or potentially rewarding, then dopaminergic systems become relevant. At the center of this is the ventral tegmental area, or VTA, in the midbrain. The VTA sends dopaminergic projections to the nucleus accumbens and the prefrontal cortex. This corresponds broadly to the mesolimbic and mesocortical pathways. These circuits are central to motivation, rewards prediction, salience, and behavioral activation. It's worth emphasizing that dopamine should not be reduced to a pleasure chemical. That is too simplistic. Dopamine is better understood as a part of system involved in wanting, seeking, anticipation, prediction, and effort-related behavior. Wanting is not the same as liking. The patient may continue to want, pursue, or seek a behavior even when the actual hedonic experience or liking is minimal or diminished. This helps explain compulsive patterns, addictive behaviors, emotional eating, and many habitual actions that persist despite reduced satisfaction. It's also important to note that behavior is not governed by dopamine alone. This is a network process. Glutamate contributes major excitatory signaling. GABA provides inhibitory and regulatory braking. Endogenous opioid systems are more closely linked to pleasure or hedonic liking. Serotonin contributes to broader modulation of mood, regulations, and stability. So behavior is not product of a single neurotransmitter. It emerges from coordinated neural networks interacting over time. After behavior occurs, the brain evaluates the outcome. This is the point at which expectation and reality are compared. If the outcomes is better than expected, behavior reinforcement is strengthened. If it's worse than expected, the relevant pathways are weakened and motivation may be declined. Over repeated cycles, this process contributes to reinforcement learning. With repetition, neural pathways becomes more efficient. The behavior becomes easier to trigger and more automatic. This is the neurobiological basis of habit formation. Habits are therefore not best understood as simple matters of virtue or weakness. They are learned efficiencies built into neural system through repetition and reinforcement. When we bring all of this together, another clinically important distinction emerges: the relationship between limbic and cortical systems. The limbic system is relatively fast, emotional, and oriented towards immediate rewards and salience. The prefrontal cortex is more reflective and more capable of long-term planning and inhibition. Behavior can therefore be understood as negotiation between these systems. The cortex may generate rational plan, but if the limbic system does not experience the behavior as sufficiently rewarding, sustaining that plan becomes difficult. This leads to one of the central propositions of this talk. Lifestyle change often fails when the cortex provides instruction, but the limbic system doesn't experience reward.